So I decided to do my project on DNA structure and replication. DNA, or deoxyribonucleic acid, is found in the nucleus of cells and functions to store genetic information and pass that information on to new cells. Each segment of DNA is called a nucleotide. So in this drawing, I've decided to display a nucleotide. In the green circle here is a phosphate. And then in the red shape here is a sugar. And a sugar in DNA is known as deoxyribose. And then in the orange is a nitrogenous base. And a nucleotide is made up of all three of these. The DNA molecule is called a double helix, which looks like a twisted ladder, as you can see in this photo. The upright parts of the ladder are made up of the sugar and phosphate molecules, and the rungs are made up of pairs of nitrogenous bases. There are 4 possible nitrogenous bases. Here in the green, I am writing adenine. And then in the pink is thymine. And in the blue, we have cytosine. And then in the tan, we lastly have guanine. The bases that make up these pairs complement each other, so adenine will only bond with thymine, and cytosine will only bind with guanine. They are held together with weak hydrogen bonds. There are approximately 6.5 billion base pairs in a cell. During cell division, the DNA in a cell needs to replicate itself in order to pass on an exact copy of new cells. Here in this drawing, you can see the guanine and cytosine bonds breaking off, as well as the adenine and thymine bonds breaking off. This is accomplished by the original DNA strand separating between the base pairs, creating 2 different templates. A series of enzymes will unzip the original DNA molecule and add the matching nucleotides along each side of the ladder. This will eventually create 2 strands of DNA which are identical to the original strand. Here you can see drawn in green is the original strand used as a template to make the complementary strands in orange.